Recent discoveries in cancer research have identified numerous cellular receptors as promising targets for cancer therapy. One such receptor is folate receptor alpha. This receptor allows rapidly growing cells to uptake folate. Folate, a B-complex vitamin, is important for cell growth and survival. Cellular uptake of folate is mediated through multiple transport systems. Folate receptor alpha is one of the folate transport proteins that can bind folate with high affinity and mediate its transfer into the cell. Dividing cells require new synthesis of nucleic acids and proteins. Folate is important for DNA replication, cell growth, and division. The expression of folate receptor alpha in normal cells is low and tightly regulated. In contrast, some types of tumor cells express high numbers of the receptor on their surface. The increased number of folate receptor alpha may be associated with tumor progression and poor disease prognosis. The surface of a cell is made of a lipid bilayer forming a membrane. Folate receptor alpha is anchored on the outer side of the membrane. In the same area, a tyrosine kinase called Lin kinase is attached on the inner side of the membrane. Lin kinase is a cellular factor that signals inside the cell to other cellular components to initiate the process of cell division and ultimately tumor growth. Farletuzumab is a humanized monoclonal antibody. Current data suggests that it binds strongly and specifically to folate receptor alpha. Once farletuzumab has attached onto folate receptor alpha, it is believed that Lin kinase is displaced from the membrane, and it no longer signals inside the cell. This event is thought to be responsible for slowing down the generation of new tumor cells. In addition, farletuzumab bound to folate receptor alpha on the surface of tumor cells may have the ability to recruit cells of the immune system like natural killer cells. It is hypothesized that these immune system cells attracted by farletuzumab could then mediate the destruction of tumor cells via a process known as antibody-dependent cellular cytotoxicity, or ADCC. Farletuzumab may also activate the complement cascade and lead to complement-induced cytotoxicity. In summary, farletuzumab may offer a multimodal targeted mechanism of action. In vitro, farletuzumab appears to have anti-proliferative and cytotoxic activities in tumor cells which overexpress folate receptor alpha. Ongoing research will allow us to shed more light on this critical pathway. Farletuzumab is currently being studied in clinical trials for certain cancer types that overexpress folate receptor alpha.